In this lecture, we're going to talk a little bit about some of the scientists who performed the experiments that not only determined what the makeup and structure of DNA is, but also how DNA ends up replicating itself. So let's begin. We've already been talking at length about Gregor Mendel and how his experiments breeding pea plants with each other helped him determine the laws of dominance, segregation, and independent assortment. And Gregor Mendel thought of genes as almost like little individual particles that were passed on from parent onto offspring, but he really had no idea what inside of the cell contained these genes or these particles, because quite simply microscopy and lenses weren't powerful enough for scientists to actually look inside of the cell and see the components of it. So when he published his work in 1866, on the laws of inheritance, folks were kind of like, that's nice. And mostly that work was almost forgotten about until in the late 1800s, we started observing things like chromosomes and cell division and what's going on inside of the cell through meiosis. And scientists like Fleming and Van Beneden had started looking inside of the cell. It really wasn't until the early 1900s with Thomas Hunt Morgan and Alfred Sturtevant who realized that these genes, these inheritable units, were actually located on chromosomes themselves. And what they actually did in order to figure this out was they had a series of traits and characteristics of fruit flies. They crossbred these flies, and then they applied Mendel's laws of independent assortment to determine whether or not the traits for different characteristics were either linked on the same chromosome or unlinked and found on separate chromosomes. Now, on top of that, once they found these genes that were linked on the same chromosomes, they realized that recombination can occur. And this way, by looking at the recombination frequency between two linked genes, they were able to actually go and map out the locations of specific genes on chromosomes of fruit flies. And this is back in the 1910s, before scientists even knew that the DNA was the component of the chromosome that actually contained the genes and the genetic information that could be passed on from one individual to the next. So, what were some of the experiments that happened after Morgan and Sturtevant that helped us figure out that DNA actually contained the genetic code? Well, important to determining this were some experiments on mice and strains of Streptococcus bacteria that we've already talked a little bit about as sort of an aside during lecture this past week. So the scientist Frederick Griffith in the 1920s was responsible for demonstrating that the genetic material from one set of bacterial cells can be incorporated into the cells of living bacteria in order to give those living bacteria new traits, new characteristics. So in this system, there were two different types of Streptococcus bacteria that Griffith was actually working with. The first strand of Streptococcus bacteria was called the S strand of bacteria, the smooth Streptococcus bacteria. Now, the S strand of bacteria, when they were living, if you injected a mouse with them, they found that this smooth strand of Streptococcus was deadly, caused the mice to become sick, and caused them to end up dying. In contrast to that, The R strand of the bacteria, the rough strand, was completely harmless to mice. So if you went and you injected them with the rough strand of bacteria, these animals would simply live. Now, furthermore, what Griffith found out that he could do was that if he were to go and heat up the S strand of bacteria and inject it into the mouse would still live. Therefore, it was something that the bacteria were making while they were alive that would be able to kill the mouse. So, if Griffith went and took some of the dead S strand of bacteria, put the harmless R strand of bacteria in that same solution that had the cell contents of the burst S strand of bacteria, then the R strand of bacteria could pick something up from the dead S strand and then turn themselves into a virulent or deadly strain of bacteria. So what this demonstrated is that bacteria can take pieces of genetic material from other dead bacteria and give themselves new traits. With this system in place, this experimental system in place, other scientists were able to then figure out what type of macromolecule was it inside of these bacteria that could transfer these traits from one individual to the next. And those sets of experiments were done by Avery, MacLeod, and McCartney. So in the 1940s, what these scientists ended up doing was they took the S strand of bacteria, they killed it, so now all of its contents were in a solution. And then what they did was they went and they would systematically, from that soup, destroy different types of macromolecules, put the R strand in the remaining solution, and see whether or not it could make that R strand virulent. So the first thing that they did was they went and took some of that solution and they destroyed all of the RNA in it. They then put the R strand in there, and what they found out was that in the end, even with the RNA destroyed, those R strand of bacteria could develop into the virulent, dangerous S strand of bacteria. Therefore, as a result, they concluded that the RNA was not the material that contained the genetic instructions to transform bacteria. They took another solution, they destroyed all the proteins inside of it. And again, when they put the R strand bacteria into that solution, they found that the R strand could still transform into a virulent, deadly form of the bacteria. It wasn't until finally they destroyed the DNA in that soup of killed S strand bacteria. It wasn't until they destroyed that DNA that they found that the R strand that you would put in that solution could not transform. Therefore, the experiments of Avery, MacLeod, and McCarty determined that it was actually the DNA that had the genetic code in it that could cause the inheritance of one trait from one individual to the next. So now that scientists have realized that it is the DNA in the chromosomes that has the instructions for different traits of individuals, they still haven't figured out what is the actual structure and what is the actual shape of DNA. There are a number of people in the early 1950s that helped determine the structure of DNA. And among those include Erwin Chargaff, Linus Pauling, Rosalind Franklin, who worked in the same laboratory setting as Maurice Wilkins, and James Watson and Francis Crick. And I'd like to thank my mother-in-law, Pat Bartlett, who was a fantastic high school biology teacher in Massachusetts that sort of helped me set up the structure for just talking about the discovery and the determination of DNA structure. So thanks, Pat, for some of the help with this. So the first individual that we've heard about at length already is Erwin Chargaff. Erwin Chargaff was born in the Ukraine in 1905. After he got his doctoral degree, he actually spent about 5 years at Yale. And then in the early 1930s, he actually held a position at the University of Berlin. However, with the rise of the Nazi Party, Erwin Chargaff was actually forced to resign because he was Jewish. And as a result, he to escape persecution, he fled Germany and actually left for the University of Columbia here in the United States shortly before World War II. And what he found in 1950 through looking at DNA, he found that the mass of adenine and thymine in any one piece of DNA or any one chromosome, usually those two masses of adenine and thymine equaled each other. The same thing he found out was that guanine and cytosine always evened out, always had the same amount of mass. So essentially, he found that the mass of purines always equaled the mass of pyrimidines in any one piece of DNA. The second thing that he figured out was that even though the mass of purines always equaled the mass of pyrimidines, the actual ratios of cytosine and guanine compared to the amount of adenine to thymine, those ratios actually differed from different individuals of different species. So what this helped stimulate the thought that the differences between species came in the differences in their relative amounts of adenine to thymine and guanine to cytosine, sort of laying the ground rule, the basis of thought for the fact that these nitrogenous bases could be the sort of instructions needed to transfer genetic information from one individual or another and make individuals unique from each other. So that was Erwin Chargaff in 1950 realized this. The next individual that we should really talk about when we talk about the discovery of DNA or any discoveries in biochemistry is Linus Pauling. Linus Pauling was an amazing scientist, and what he helped determine, he's sort of the father of biochemistry. He determined the different rules and that sort of govern specific angles in bonds that were created between atoms within a molecule. So by establishing these basic rules of bond angling, you could determine the shape of different molecules. And of course, because the shape of a molecule determines its function, this is a very important thing to know. In 1951, he published a paper in which he described the alpha helix shapes, those twisted helix shapes that are found in polypeptides. And as a result, in 1954, he won the Nobel Prize in Chemistry for the nature of bond angles and bond chemistry, in addition to discovering the alpha helix shape of polypeptides. Now, in the early 1950s, about 1952 and 1953, shortly after he published on alpha helixes, he started to turn his attention towards determining the structure and the shape of DNA, which actually got a lot of people who were already studying the structure of DNA really nervous, because everyone was convinced Linus Pauling was going to figure this out before everyone else did. So it kind of lit a coal under everyone else's butts to really get them to work a lot harder and start publishing some of their work. He unfortunately, he did not determine the correct structure of DNA, although he had some hypotheses on these that turned out to be incorrect. But he went and actually turned his attentions towards raising awareness for the elimination of above-ground testing of nuclear weapons. And in 1962, he actually won the Nobel Peace Prize for that. And he's one of only 2 people to ever be the solo winner of 2 different Nobel Prizes in different categories. One for chemistry, and then he won one for the Nobel Prize in Peace. So really an incredibly amazing individual. However, he did not actually determine the correct structure of DNA, but influenced many others who did figure it out. The next group of individuals that were really important in laying the ground rules of determining the structure of DNA were 2 scientists who worked in laboratories in King's College in England. Their names were Maurice Wilkins and then Rosalind Franklin, who worked in the same department as Maurice Wilkins, but did not actually work under Maurice Wilkins. She ran her own individual lab. Maurice and Wilkins and Rosalind Franklin were very important in determining the structure of DNA because Maurice Wilkins developed important techniques in order to crystallize DNA and help us determine what its shape was, while Rosalind Franklin was an expert on a technique called X-ray crystallography, which was a technique that allowed you to essentially take a picture of DNA. Now, both of their works, particularly Rosalind's, helped create the foundation in which other scientists realized that DNA actually forms this sort of spiral, twisty helix in shape. And they were very important scientists working on determining the structure of DNA. The other scientists that were working in England also at Cambridge University was an American by the names of James Watson and the scientist that he worked with, which was a physicist, an Englishman by the name of Francis Crick. At Cambridge University, they went and they proposed that the shape of DNA was a double helix and primarily worked this out by constructing models, 3-dimensional models of what the structure of DNA should actually look at. And what they were able to do was they took inspiration and built off of discoveries of other individuals. So, for instance, from Erwin Chargaff, they knew that they figured that adenine would always have to bind with thymine, and guanine had to bind with cytosine. They knew what angles these atoms would have to form in creating this structure because Linus Pauling had already determined what are the bond angles that occur between different atoms within a molecule. They also went and realized that there should be some sort of twisted helix shape, and they determined that off of the studies and the workings of folks like Maurice Wilkins and Rosalind Franklin. And the really big idea that not only did they synthesize all of these ideas together to come up with this 3-dimensional double helix shape, but they also proposed the anti-parallel structure of the 2 strands of DNA that are linked to each other in the helix format. So really, they did a fantastic job of sort of synthesizing all of the work from other scientists. So in the end, who gets credit for discovering the double helix shape of DNA? Well, I'd hate to say, in a sense, it kind of gets political. So this is an interesting story. In early 1953, Linus Pauling's son gets a draft version of a paper that Linus Pauling is going to publish about the structure of DNA, and he shares it with Watson and Crick. In this draft, Linus Pauling suggests that DNA actually has 3 strands, and Watson and Crick realize that this is totally wrong. However, they realize Pauling is probably getting pretty close. So James Watson travels off to King's College to talk with Maurice Wilkins and Rosalind Franklin about the 3 of them collaborating and working on publishing and determining this 3-dimensional structure of DNA. On this trip, he offends Rosalind Franklin, and as Maurice Wilkes is trying to sort of calm him down a little bit, he actually shows him a sneak peek of a picture, the photo number 51, that Rosalind Franklin took of the 3-dimensional shape of DNA. So it gets Watson realizing that, hey, there's a helical shape. Now, about 1 month later, Rosalind Franklin is in the process of leaving King's College for another university, and the head of the division at King's College, and that's working with these laboratories, decides that all of her photos and all of her notes have to stay at the university anyway, so Maurice Wilkins actually ends up getting a copy of all of these papers anyway. So this, of course, spurs on Watson and Crick to relatively quickly develop their model, their 3-dimensional model of DNA as a double helix that has an anti-parallel structure. And in the meantime, Wilkes and Wilkins and Rosalind Franklin are also writing their papers about the 3-dimensional structure of DNA, which they also describe as being a double helix. So as a sort of political concession between the 2 universities and these 3 groups of researchers that are developing a picture of what DNA looks like, they decide that in the scientific journal Nature in 1953, they would publish 3 papers, each of them describing the 3-dimensional structure of DNA. The first paper published in order is Watson and Crick's. Then the paper is published by Maurice Wilkins. Then the third article is going to be Rosalind Franklin's article published in Nature. So as a concession, they publish all 3. However, some people reading it, seeing that Watson and Crick's article appears first in the journal, kind of has the connotation that the other 2 articles are more supporting of their major work. In the meantime, unfortunately, in 1958, Rosalind Franklin dies as a result of ovarian cancer. 4 years after Rosalind Franklin dies, the Nobel Prize Committee awards the Nobel Prize in Physiology or Medicine to those who discovered the structure of DNA, which happens to be James Watson, Francis Crick, and Maurice Wilkins. Unfortunately, the Nobel Prize is never awarded posthumously. There you go. And since Rosalind Franklin had died in 1958, 4 years earlier, she could not be awarded the Nobel Prize, which is really a very unfortunate, and sad event, especially since in the years afterwards, the role of Rosalind Franklin was quite honestly very much downplayed in this amazing discovery. But fortunately, in the recent decade or so, more and more people have come to appreciate and fully realize Rosalind Franklin's incredible contributions to the discovery of the 3-dimensional shape and structure of DNA. So now by 1953, we understand what the structure and shape of DNA is, and now we're trying to figure out, how does DNA actually work? One of the major ways, one of the major findings of how DNA works is, how does it actually go and replicate itself? Now, Watson and Crick had hypothesized that DNA replicates semi-conservatively. However, it wasn't until there was another pair of scientists that actually came around and performed the experiments to determine and to prove that DNA replicates through in a semi-conservative manner. Those two scientists were scientists by the name of Michelson and Stahl. Michelson and Stahl determined that DNA replicated semi-conservatively through the use of stable isotopes. Now, we remember way back in the first month of BIOL110, we talked about an isotope being a form of an element that has a different number of neutrons. The isotopes that they used were nitrogen-15, which is the heavy version of nitrogen, a stable isotope, and then nitrogen-14, which is a relatively lighter version of that atom. So what did they actually do? Michelson and Stahl went and they took a broth that was filled with heavy nitrogen, nitrogen-15, and in that broth of heavy nitrogen, they raised bacteria cultures over generation, over generation, over generation, until eventually they got to a point where all of the DNA, all of the nitrogen found in the DNA of those E. coli, of those bacteria, all contained that heavy nitrogen-15. So when they would go and they would weigh those bacteria, or the DNA from that bacteria, they would find that all of the DNA from that bacteria were of a relatively heavy weight because they had nitrogen-15. So here, down in the bottom of the graph, you'll see an example of a drawing of a double-stranded DNA that both strands are built with nitrogen-15 in blue. Okay, so what they did next was they took all of the bacteria that had that was labeled with only heavy nitrogen-15, and then they went and they grew them in a solution of lighter nitrogen-14. So that way, whenever they would replicate, any new strands of DNA they would make would be relatively lighter because it would be made with nitrogen-14. They kept that bacteria in that solution for about 20 minutes, which is about how long it takes for all the cells to go in, replicate only one time. And what they found out as a result was that all of the DNA that they collected from those bacteria was of a slightly lighter variety of DNA, and it was slightly lighter than the previous generation because it had semi-conservatively replicated, and as a result, all of the DNA included one strand of heavy DNA from the template, from the original, and then its complement was the nitrogen-14, which was slightly lighter. They took that same culture, that generation of bacteria, and they kept them in nitrogen-14 again for another generation to go through one more round of replication. After one more round of replication, they found two varieties of DNA. Half of the DNA was of that intermediate weight. That intermediate weight was the DNA that had one strand of nitrogen 15 and one strand of nitrogen 14. The other half of the DNA was even lighter than the ones they had made before. Those were actually consisted of two strands of DNA that was made with the lighter isotope of nitrogen, nitrogen 14. So what happened was they deduced that what must have happened was, as the original DNA had replicated from the previous generation, as it split, each one of those had created one strand that had the nitrogen 15 template and a nitrogen 14 complement. In addition, the other strand that was originally nitrogen 14, when it made its complement, it had made a complement made of nitrogen 14 also, making it much lighter than that. So you can see by looking at the different weights of the different molecules of DNA that were made, Michelson and Stahl were able to use these stable isotopes to determine that, to have empirical proof that indeed DNA replication was semi-conservative, as Watson and Crick had originally proposed. So, one thing that I really hope that you get from this lecture is that you realize that, these scientists who made these discoveries and performed these experiments, I mean, they were real people too. They had successes, they had failures. Some of them could be incredibly competitive and want to scoop somebody else and figure this out before they were. Some of them were completely selfless. And in the case of Rosalind Franklin, right, put her life on the line to actually work with things like X-rays to make their discoveries. They were really they were just regular people, right, and really interesting people. And I really hope that all of you have a chance to go and take a look at in 2005. There was a TED Talk given by James Watson from the famous Watson and Crick. The YouTube video to this is below, and even though it's about a 20-minute watch, it's really a fascinating sort of interview, because you really see all of these personality traits, within this person. And he talks about, the colleges he didn't get into, and the women who refused to marry him, and his in addition to all of the great successes that he had. And you actually do kind of look in and see some of the maybe unprofessional and somewhat not particularly complimentary views that he has on some of his contemporaries. So please, I strongly encourage you to check that out. And he's a really entertaining speaker and actually really pretty funny also. And if you're interested in learning more about Rosalind Franklin, there's a great biography of her life called Rosalind Franklin, the Dark Lady of DNA, written by Brenda Maddox. And I hope this gets some of you to start thinking about, these people just not as names in textbooks, but as full, complete, human beings. So hopefully you enjoyed this lecture, and I will see you all in class next week. Thank you.